Thank you for the introduction and thank you for the land acknowledgement. I forgot to include it, so I'm very glad that you mentioned that today. It's very important. As mentioned, my name is Veronica. I'm from the School of Biomedical Engineering. I'm doing my master's degree, technically on my way out. I'm graduating next month, which is a bit sad, but that is my field of specialty. The title of my webinar today, as Sam said, is Imaging Blood and Clot Interactions on Biomaterial Surfaces, an experiential approach to SEM imaging. I say experiential because much of what I will talk about is drawn from my own limited experience with SEM in my master's research. If at all you want to contact me or connect on LinkedIn, I'd be happy to get to know you. I've provided my email here and you can just look me up on LinkedIn if you'd like to. All right. So before I jump into my research and more of the methodology, I want to just present an overview of electron microscopy in case we have any viewers who have never used it before or new to the field. From a historical perspective, briefly, scientists have had a fascination with the microscale for centuries. With the emergence of applied optics in the 4th century, the first microscope in the 1600s, and then electron microscopy in the 1930s. It was invented by scientists Knoll and Ruska, which is very interesting. Our understanding of the cellular and atomic structure of biological systems has evolved and played an important role in the development of science. So our knowledge of the microscale makes up the building blocks of modern scientific theory. So it's very important to acknowledge the usefulness of these imaging modalities. The electron microscope is a specialized imaging tool. It leverages a beam of electrons and a series of apertures to generate high resolution grayscale images. Two general types, very broadly speaking, are TEM and SEM, very different modes of action. TEM generates very detailed images of the internal structure. For example, the herpes virus, this is a very nice image with the capsular structure, sort of looking at the internal structure. And SEM generates very nice topographical images. So looking at the scattered electrons to generate this topographical image. So E. coli bacteria with its characteristic rod shape, which is very informative as well. In my research, I was more focused on SEM, especially for imaging blood cells, thrombus, things like that. So this chart really captures the utility of EM at various size scales. And it really shows us that EM is not only useful for gaining insight into the shape and size of biological structures, but also structure and function, which is something I'm so fascinated in. It's such an important concept in biology. So looking at the human hair, for example, these keratinized layered scales are associated with the strength of the hair cortex, which is interesting. We gain insight into that. Likewise, the papillary architecture of the lotus leaf is associated with its water repelling property. So really looking at the appearance of these images gives us a great insight, not only to shape and size, but also into structure function. In essence, giving us a look into the how and why in scientific inquiry, and which is critical for all science, I would say. So not surprisingly, and this is mostly for interest, EM has a variety of applications, and some of you may work in different fields, so I thought I would include this here. So the list is not comprehensive, but material science, definitely for quality control, characterization of semiconductors, microchip elements, which is very cool, very high precision. So high resolution images are required. Geological science, something I didn't know, but identifying minerals, looking at gemology, medical science, a very important field, large number of applications. Pathology is a great example, especially renal pathology. Also virology, so identifying novel viruses. One of the first and most interesting images that I've seen during COVID was that capsular structure with the club-shaped projections on the virus. And That in part was identified using SEM, so very interesting application. And also in research, a very important area. I'm sure that's why you are here. You use EM in your own research. So we can gain insight into body implant interaction, for example, thrombosis or integration of bony implants, whether let's say it's a knee replacement or a hip replacement, really looking at how well that implant integrates with the body. And also platelet biology and thrombosis. SEM has played an important role for over 50 years in this field, giving us insight into how platelets function, different platelet structures, and what this means from a functional perspective. So from now on in this presentation, I'll be sort of zooming into this quadrant, mainly from a research perspective, as this is a fundamental part of my project. So a bit about my work. My thesis was developed in Dr. Tauhid Didar's lab for biologically inspired engineering. And this was in collaboration with Dr. Jeffrey Weitz at the Thrombosis and Atherosclerosis Research Institute. And I was given this challenging objective, which was really, we want to create a testing microenvironment at the benchtop that allows us to study body implant interaction in a physiological like testing scenario, which is very difficult to achieve in vitro, especially if blood flow, for example, is an integral part of that environment. So I had to develop a platform that incorporated vascular prosthetics, so heart valve, vascular graft, vascular stent with blood flow. I did this with microfluidics, as you'll see later on in this presentation. And then the second objective was to apply this platform to quantify the thrombogenic performance of different surfaces and materials, really zooming into that interaction between blood and the surface. And SEM was an important part of that. And we will cover that as well as some methods that I used. So just to provide a background in case this is not your area of expertise, the motivation for my research on the most fundamental level is cardiovascular disease. So cardiovascular disease is a very serious condition that is widespread. You may have loved ones who are affected by cardiovascular disease. And it is the leading cause of death and disability worldwide. An estimated 1 in 12 Canadians has some form of CVD. And this incurs a financial burden in the healthcare system of over $22 billion every year. So this is a huge magnitude of problem, affects a large number of people. So it's very important that we try to develop therapeutic approaches that can resolve this issue. So CVD is an umbrella term. It describes dysfunction of blood vessels in the heart. This could be a range of conditions. The common denominator here is that most of these conditions arise from the narrowing and thickening of small and medium diameter arteries due to atherosclerosis, which is the buildup of fatty deposits and fibrotic plaque. The prevalence and cost of this condition has driven the development of therapeutic solutions. The use of drugs like statins, fibrinolytics, and blood thinners is often used as a prevention and early stage intervention. But when this is inadequate, the use of vascular surgery and vascular prosthetics is required. This may involve heart valves, vascular stents, vascular grafts. These are made of a range of materials such as PTFE, PET, Dacron, stainless steel, just a range of non-biological materials. And these are often implemented in different types of surgical operations. If you're interested, it's a very interesting field, I would say. So angioplasty, atherectomy, bypass. It's important to mention that although these devices are an important part of the therapeutic approach for people with cardiovascular disease, they do not always perform well in the body. Thrombosis of vascular prosthesis is an ongoing issue in clinical practice. It not only undermines device function, but also introduces risk to the patient. So in some cases, if it's a peripheral complication, This can result in limb loss. This can also result in death. So this is another problem in the cascade of problems presented by cardiovascular disease. I've provided a series of images here to really show you what thrombogenic failure could look like on these devices. This is a heart valve, mechanical heart valve, a coronary stent, and vascular graft. As you may infer from these images, the surface or lumen of these devices becomes fouled or encapsulated in a red fibrous matter. This is what is known as a blood clot or a thrombus. It's important to mention that thrombosis of vascular prosthesis is a complex event. You may ask, why does this occur? There have been a series of explanations developed and postulated over time, but one of the most prominent approaches to explaining this phenomenon is Virchow's triad. So this was developed by a scientist centuries ago named Rudolf Virchow, and he claimed that thrombosis of vascular prosthesis arises to three essential pillars. The first being the presence of blood flow, especially altered blood flow conditions, so low flow conditions and stasis, the presence of surface, whether this is a vascular injury, whether this is a foreign material, it's an altered surface. So it's not a native blood vessel, it's a foreign material or a disrupted biological material. And also hypercoagulability. So in my case, I was not interested in the third pillar, it was a bit out of scope, but the first two pillars were essential when I was considering how to develop this platform I was tasked with in my project. It is known clinically that these two pillars are essential in prosthesis performance. A great example of this is looking at lower extremity bypass involving vascular graft. This is a context where low flow conditions are predominant, and also with synthetic materials like PTFE. So this combination of two undesired variables does result in poor performance outcomes. This is reflected here in this patency curve. So comparing a PTFE graft, patency or openness versus a native biological graft. So, and this is in a lower extremity context. So clearly, thrombosis of vascular prosthesis is a significant issue. It is especially an issue in low flow contexts with synthetic materials like PTFE. So that was kind of the focal application in my research, looking at PTFE as a material of interest and looking at this low flow condition as our flow variable of interest. So in terms of the first pillar of Virchow's triad, I'm not sure how much background you have on this, so I thought I'd provide this diagram. So the presence of altered surface, like I mentioned, could mean a foreign material like a metal stent. It could mean PTFE. It could also mean a disrupted blood vessel. And depending on which surface is present, two distinct pathways in the coagulation cascade can be triggered. Here, the coagulation cascade refers to a series of proteolytic reactions that converge to form the common pathway, starting at this green chain here, which ultimately generates fibrin, which is the backbone of a blood clot. So fibrin is the polymerized web-like structure that arises from the plasma protein fibrinogen. In my work, we were mostly interested in the leftmost branch of the cascade, looking at the interaction between the synthetic substrate and blood. And we were mostly interested in the end point, so bulk clot formation, rather than individual steps in this cascade. But definitely, this was an integral part of my research and required a thorough understanding. It's important to mention as well, like I mentioned a few slides ago, based on Virchow's triad, the presence of altered flow is also an important consideration when we predict or try to understand why some prosthetics perform well and why some don't. So the presence of blood flow really gives the process of blood material interaction a spatial and temporal dimension. For example, in the early stages of blood material interaction with a vascular prosthesis material like PTFE, the adsorption of plasma proteins, platelets, and cells is altered by local blood flow conditions. So this can affect the rate of delivery and removal of clot-forming elements, for example. It can also result in shear activation of proteins like von Willebrand factor and platelets, conformational dependence, which can accelerate clot formation and render certain materials or implants more prone to failure. Clearly, it is a combination of surface and flow factors that do govern the performance outcomes. And for me, this was very important consideration in the development of my testing platform, because I wanted to generate this physiological-like testing environment so we can gain pretty realistic insight into the performance of these materials at the benchtop. Exactly. So you may ask, how can we achieve this? And it is something I was stumped on for a very long time. So I decided to translate methods from microfluidics. So microfluidics, they harness micro scale channels to generate fluidic profiles that mimic vascular conditions. This is usually by isolating and recapitulating wall shear stress. There is a standard and well-established formula for calculating wall shear stress in this dimension of channel. So I drew inspiration from existing literature to translate channel geometries to create this physiological-like vascular flow condition, wall shear stress, and additionally incorporated a prosthetic material. I achieved this using a sort of unique bonding method. And this ended up being a very versatile approach that very successfully captured those two pillars of Virchow's triad, not only the presence of surface, but also the presence of flow. And both of those variables could be tailored depending on your investigation of interest. So I called this a vascular graft on a chip, which is quite interesting. All right. So once I developed this platform, I started to think about, well, what can I use it for? What investigations could I perform? And although this was an incomplete module of my research, I thought I would include it here for your interest, maybe spark some ideas, inspiration in your own work. So as I mentioned a few slides ago, the coagulation cascade is a very important part of blood material interaction, especially adverse blood material interaction. And the adsorption of plasma proteins really triggers this process that culminates in clot formation. So I wondered if we could use this platform to study protein adsorption in real time, and specifically if we could leverage fluorescence to gain insight into the adsorptibility of different prominent blood protein species like albumin, fibrinogen, factor 12. So I performed this very simple assay, channels in parallel, looked at relative differences in adsorption, and it was pretty informative. This was a pretty interesting experiment, very insightful. Not quite a complete story at this point, but very interesting application nonetheless. In terms of what I can suggest based on my experience, I would say studying protein adsorption is a very important aspect, especially if you're developing a new material or new coating. And based on my experience, I would say fluorescence can be an informative measure of protein adsorption. It can be difficult to interpret. It's more of a macro scale observation. And I've also read that fluorophores can affect adsorptibility of proteins. It can alter the isoelectric point of proteins. So you'd have to be really careful and thoroughly investigate whether your fluorophore is appropriate for your investigation. But in addition to fluorescence, I would suggest using a combination of techniques. So not only gaining this very qualitative insight, but also a quantitative insight. So you could use quartz crystal microbalance or QCM, atomic force microscopy or AFM, radio labeling. You can use an assay like BCA. There are a number of approaches, and it's good to use a combined approach, I would say. So And SEM can be informative for protein adsorptibility. Again, we have this issue of interpretation. It's very difficult to say which species have adsorbed here. What exactly is this layer here? If you had a purified solution or purified perfusate with a single protein species, interpretation could be easier, but in general, it could be quite challenging. So to sort of avoid this confusion, I would suggest using a combination of approaches such as the ones I've listed here, and really gaining as much data as you can, very diverse data, and really construct a very strong and coherent story. All right. I found this really interesting table here, and it was actually co-authored by Dr. Ryan Wiley, who is at McMaster, and it really concisely illustrates what type of species we should test for or assay for on our surfaces, depending on the implant context as well as the duration. So if this is something that could be of use to you, I definitely suggest reading this paper. It was in Frontiers in Chemistry, published in 2020 by Jesmer and Wiley. So for example, in the short term for blood contacting prosthesis, fibrinogen is an important consideration, macrophage perhaps important consideration, looking at bulk clot formation. This is all very important, and again, this is a very informative table, so I really recommend looking into this paper if you can. It also has a good collection of methods you can use to quantify surface performance. So this is very good, and thanks to Dr. Wiley. Important to mention, thrombosis of vascular prosthesis is a significant issue. We saw this. It undermines device function, also introduces complications for the patient. So not surprisingly, scientists have worked very diligently to find approaches to overcome this issue. And one such approach is the use of coatings. Development of antithrombogenic coatings is a very active area of research. There are many labs at McMaster that study this, my lab included with Dr. Tauhid Didar. It is estimated that between 2008 and 2016, $40 million worth or more of grants were awarded for the development of novel antithrombogenic coatings. From this, the use of bioactive, biomimetic, bioinert, micro nanostructure coatings have been well described. Some have been deployed in clinical settings. Pyrolytic carbon-coated heart valve is an example. Heparin-coated vascular graft by the company Gore and Gentige are other examples. There are many others. There are many others for sure, especially in literature. These coatings work to suppress protein adsorption, blood clot formation, and promote desired biological processes, especially with biomimetic coatings. For example, incorporating an endothelial capture antibody like CD34, it really promotes endothelial integration, which is desired. So we regenerate that native endothelium, which has innate antithrombogenic properties. So again, this is a very important area of research that hopefully in the future will help to overcome this issue of thrombogenic failure of vascular prosthesis. As a side note, if you are developing a novel coating, which again is an important area of investigation and in the research lab I'm in, you may wonder, how can you study these surfaces? How can you qualitatively and quantitatively assess these surfaces? Fortunately, SEM is a very useful tool for this purpose, especially if you're dealing with a nano microstructured surface, like a hierarchically structured surface. There's a student in our lab, Shadman Khan, who developed this technology. Also nanotubules, which another student in our lab, Leanne Ladouceur, developed. So SEM is very informative. It helps us visualize what these surfaces look like in a very qualitative sense. However, as usual, I would suggest using a combined approach that integrates not only this qualitative observation, but also a quantitative observation. So for example, using interferometry or AFM to gauge the roughness uh, in a numerical language, or the thickness of your coating in a numerical language. This is very insightful and can help with the optimization of your coating in the development process. In general, I would suggest using a combination. SEM is a very good first step. Once you've sort of grasped the topography of your surface, then dig a bit deeper and really search for those quantitative metrics. In my project with this developed platform, because it was very easy to run test conditions in parallel, we thought about implementing distinct coatings in parallel and really viewing this platform as a translational tool, something that can allow us at the benchtop to simulate performance and screen the performance of novel antithrombogenic coatings. So this was one module of my thesis. We implemented a novel antithrombogenic coating called a lubricant-infused surface in parallel with unmodified surfaces, PTFE, and we assessed the thrombogenic performance of these surfaces in parallel. So it's a very interesting application, very informative, I thought, and very efficient. So very briefly, this was achieved using liquid infiltration of the coating agent, as well as a series of conjugation steps using EDC NHS with an endothelial antibody for our third distinct test condition. This was an excerpt of my thesis. So it really demonstrates here that we have our distinct coded conditions in parallel. We assayed the thrombogenic performance in parallel in real time over a series of time points. And the two markers here are fluorescing in distinct colors. The green color indicates fluorescent fibrinogen. Of course, fibrinogen is cleaved to form fibrin, and this is the backbone of the blood clot. And blue is indicative of thrombin activity. So this is a fluorogenic thrombin substrate called Boc-Arg AMC. The fluorophore amidomethylcoumarin is cleaved by the enzyme thrombin and generates this blue color, whose intensity is proportional to the extent of thrombin activity. So it gives us sort of a structural indicator as well as a functional indicator of the extent of clot formation on these surfaces. Of interest, leveraging microfluidics and wall shear stress, we applied an arterial wall shear stress, such as what would occur in a lower extremity bypass, which is a failure-prone scenario in a clinical setting. So basically, in short, we did this relative comparison. We found that the surface enhanced, the coated surface, had decreased fibrinogen adsorption, decreased thrombin activity, cumulative thrombin activity, and decreased surface adhesion. And this third conclusion was drawn primarily from SEM, which told us a lot of information, not only about bulk clot formation, so adherence on the surface, but also which types of species were adhering to the surface. It just so happens that lubricant-infused surface has a non-specific repellency property. So it generates this lubricous interface, highly repellent interface. It's a bio-inspired technology inspired by the pitcher plant, actually. So this really corroborated pre-existing knowledge that the surface is a highly repellent to a number of blood species, such as white blood cells, red blood cells, and platelets. In terms of assessing thrombogenicity on surfaces, again, I'm by no means an expert here, but based on my own limited experience, I would say there are a couple of things that you should look for. Bulk clot formation is a great baseline indicator of how a surface performs. SEM is a very useful tool for that. You also get a beautiful image. If you're lucky, you can capture important blood species in a singular image. Also, clot mass can be an informative measure. You may also wish to probe platelets and thrombin, which are important indicators in clot formation. So with platelets, you can do radio labeling, Western blot, antibody conjugation for CD41, 61, 62, calorimetric assays, Thrombin is a very useful dynamic measure. So thrombin generation assay with those fluorogenic compounds or calorimetric compounds. Thrombin antithrombogenic thrombin complex or TAT is very useful. ELISA chromogenic assays. There are really a lot of options here. I would highly suggest consulting with literature to identify your assessment method of choice. But I really want to convey that it's best to use a combined approach. So doing your preliminary analysis, SEM is a very informative tool. But again, combine this with other assessment modalities. Enrich your study with a diversity of data, not only qualitative, but also quantitative. This will really help you build a compelling story, coherent story. So I think there are a number of approaches here you could use, and I highly recommend reading more about them if this is something you're considering. I thought in the conclusion of this presentation, I would provide a handbook based on my experience as a master's student. What are sort of the main takeaways that I would want to share with others? So in terms of preparing samples, fortunately at McMaster at the CCEM, we are very lucky to have a very helpful staff. So Marcia Reed was very helpful for me throughout this project. But in general, it's good to have a baseline understanding of the preparation of your samples, although you will be assisted, which is a relief. In terms of fixation, in my protocol and sort of the standard, or what appears to be the standard in literature, using glutaraldehyde, sodium cacodylate, a post-fixation osmium tetroxide step, dehydration with an ethanol series, drying with the critical point dryer, coating with gold palladium, platinum, silver, chromium sputtering. I believe at McMaster at the CCM in the hospital, we have gold sputtering. So this is a good piece of information to prepare yourself, familiarize with the protocol. In terms of interpreting samples, this is very important as well. If you are not familiar with the morphology of the blood clot, it is possible you can draw some misleading conclusions. So make sure you can identify a red blood cell. A red blood cell has this disc-like, donut-like shape. It's relatively large. It should have this sort of spherical and round shape. If this is not the case, it may indicate there's an issue with your solution preparation. There is an issue with your protocol. So you want to be very aware of that and try to confirm what it is you're observing. Platelets are other species which are of interest in many studies. These blood cells have a characteristic shape, sort of a disc-like shape, but not impressed in the middle. And they can also be activated, in which case they adopt this dendritic-like structure. So again, easy to mistake this for a red blood cell if you're not familiar with the size classification as well as the shape of these cells. Fibrin is pretty self-explanatory. It's a polymerized web, so sort of like a spider web. It should be easy to spot. If this is not present in your image, it could mean you did not give adequate time for the clot formation to occur. It could mean there was a problem with the recalcification of your citrated blood sample if you used a citrated blood sample, or it could mean you had a defibrinated plasma sample. But this is something you should know ahead of time when you collect your perfusion sample or your assay sample. And finally, white blood cells or leukocytes. They look sort of like furry spherical cells, similar size to red blood cell, pretty easy to identify a difference. But as you'll see in my next slide, these cells can be mistaken for a type of red blood cell called an echinocyte, which is like a spiky red blood cell. So in terms of other applications, I really want to inspire you to think outside the box and recognize that SEM is useful not only for imaging blood material interaction, but also imaging thrombus structure on its own. So looking at the composition of the blood clot, the relative proportion of fibrin and platelets can tell us whether it was of arterial or venous origin. This was a very interesting paper I read. So arterial blood clots or thrombus are typically called in lay terms a white thrombus, so composed primarily of platelets, not much presence of red blood cells, whereas venous there's a fairly high density of red blood cells. And depending on the composition of the blood clot, we can gain insight into, again, the origin, the flow properties in that location, wall shear stress, whether there was some sort of pathology that occurred, like an aneurysm. In addition, we can gain insight into many disorders based on the morphology of red blood cells. So, for example, schistocytes are these disfigured, fragmented looking red blood cells. They can indicate anemia. It can also indicate hemolysis of mechanical heart valves. Sickle cells are collapsed red blood cells. And as you may be familiar with, this could indicate sickle cell disease. Or Burr cells or echinocytes are these spiky red blood cells. This could indicate that there's an issue with the osmolarity of your buffer, which is sort of an operational issue. Or it can indicate there is a pathology, so uremia or chronic kidney disease from that donor. I just want to point out here, this is something I struggled with. These cells in particular, echinocytes, look a lot like white blood cells. If we go back here. So you really need to study the morphology closely. And if you suspect it is an echinocyte, it is likely not a problem with the donor. It is likely a problem with your protocol. So make sure the pH of your solution is correct. Make sure the osmolarity of your solution is correct, whether that's your buffer or dilutant like HEPEs. This is a very important consideration. This is a trap I fell into, so I warn you now. So that concludes my presentation. I hope it was informative and interesting for everyone. I just want to provide a personal insight about research. So when I started Masters, I had no research experience. It was a very daunting challenge for me. It was very challenging on a personal level as well. And I would encourage everybody to view this challenge in a positive way. And I think this quote by Michelangelo is very inspiring. So if you imagine yourself as a sculptor, you're given a block of stone. There is a masterpiece, a statue in that stone, and it is up to you with your chisel to really reveal that masterpiece. And I feel as researchers, that is kind of our experience. We're given a research question. It may not be clear. It may be a very challenging and daunting task, but it is our job to really reveal the answer or reveal the masterpiece. So I want to thank my supervisory committee, Dr. Tohi Didar, Jeffrey Weitz, and Dr. Ravi Selvaganathapi, very difficult to pronounce, as well as my second home at TARI, and of course the support of the CCM, Marcia and Samantha. I really thank you for this opportunity. It's a great opportunity to develop my presentation skills and share my work. I'm very appreciative. And here's a screenshot of my lab. So I thank all my lab members for their help throughout this process. So that concludes my presentation. I thank everybody for their attention. And if you have any questions, please let me know, whether it's a personal question about a research journey as a grad student or about the subject matter of my research, I'd be happy to discuss. But if not, I'm glad you attended, and thank you very much.
Great, thank you, Veronica.
Oh,
that was a very interesting presentation.
Oh, thank you.
I learned a lot. I am not a biologist other than what I learned through Grey's Anatomy and various other fictional TV shows. So well, I found it very interesting.
Oh, I'm so glad. I mean, to be honest, my background was physics initially, so I didn't know anything about blood when I started this degree, and it's been a very insightful journey. So I just really wanted to share that with others who had a similar experience. Yeah.
That's amazing.
Oh, yeah.
So there are a couple of questions, and if anyone else has any questions, please place them in the Q&A, or if you're having trouble finding the Q&A, you can put them in the chat as well. So Catherine Granfield has asked she said, great talk.
Oh, thank you.
Would it be of interest to view your clots in a more active hydrated state in the SEM? And have you considered options for this, such as cryo SEM or ionic liquid coatings?
So in my case, I think my study was quite elementary. It is definitely of interest to pursue those avenues in the future. I think more information could be garnered from that type of investigation. But for me, again, there was a lot of focus on the development of the platform, and it was not really a specific focus too much looking into the morphology and the properties of the blood clot. But for sure, in the future, I'd like to sort of zoom in on that aspect and deploy those kind of technologies. And it's really interesting. So I thank you for mentioning those. Yeah.
And another question, did you have to worry about damage to your clots by the electron beam? For example, did you need to use low acceleration voltage, or was the preparation protocol able to produce quite robust samples?
I would say for me, I didn't have too many complications in the execution of the imaging. I don't think I used a particularly fragile protocol with the imaging itself. Really, the only issue I encountered throughout this experience was the presence of echinocytes, which were these spiculated red blood cells, which did not arise from the donor. It arose from my protocol. So I would say, I think my preparation protocol generated fairly robust images. And it was not really the imaging itself that caused issues. It was more my buffers. And I think as long as you execute the preparation properly, you shouldn't have any issues. But of course, don't take my word for it. Look into literature and confirm. But that is my impression anyway.
Great. Those are all the questions. So thank you so much. I encourage anyone, if they have further questions after the presentation, to please follow up with Veronica, or if you're having trouble getting in contact with her, send me a message and I can shoot an email off to her.
Yeah, I'll just pull up. Thank you. I'll just pull up my email address again in case anybody, sorry, there's so many slides here. Here we go. Here, almost. All right. Yes. So if anybody has questions, I'd really love to hear from you and learn more about your research as well, because again, I'm not an expert here, and it's always interesting to learn more applications. So feel free to reach out to me via email or LinkedIn, and I'd be really excited to talk to you.